Welcome to another episode of Till the Bottom. In this episode, we mull over the origin of life. To discuss this intriguing topic, we have with us Dr. James Saenz, a specialist in this field. James has his own research group at BCube, Center for Molecular Bioengineering in the Dresden University of Technology, where he explores how simple membranous units allow for complex biochemical interactions, thereby understanding how early life may have existed. We firstly ponder about what is life. Then we discuss the different possibilities on how life might have originated on Earth several billion years back. We go on to discuss how that may have led to complexity in living organisms and whether that is an inevitable direction life heads towards once the initial bottlenecks are overcome. Finally, we ponder if life exists elsewhere in the universe, and if so, how prevalent it is and how complex it may be. Gear up for an engaging discussion. Let's start talking.
Welcome to another episode of Till the Bottom. We are your hosts, Mitra and Nacho, and today we have for you here Dr. James Saenz. He's a group leader in BCube here in the city of Dresden, and he's researching something related to the origin of life, which is basically focused on biological membranes, right? Since this time, the conversation we're going to have is also very closely related to what you actually do, professionally speaking. So I think it would be nice to start with you providing us with a little bit of your background about your career and what you're doing at the moment. And from there, we will try to tie that, which is actually very closely related to the main conversation topic that we have for today. And we will start talking about everything related to the origin of life, and let's see how deeply we can dig into this topic. So, how did you come to do what you're doing?
Yeah, so I mean, what we're doing now is we're trying to understand how biological cell membranes work, basically. And I came here, and we work on very simple bacterial organisms, these model systems to study that. But I came here kind of through a bit of a different route than normal. So originally in my undergrad, I studied geology because I'd always been really interested in the history of life and evolution, and I wasn't quite energetic enough for the pre-med track. So I
it's a different route.
Yeah, so I started with that, and then for PhD, I went to a program that was the topic was chemical oceanography, but I was working on chemicals in ancient marine sediments. And in particular lipids. So the subfield that I was in was interested in trying to detect lipids from dead cells that were preserved in really, really ancient sediments. To use them as kind of like molecular fossils to reconstruct ancient life.
And by ancient, you mean how many?
Well, really ancient is like billions. And then, but there's a whole range of people in that field that study stuff from thousands to millions to billions. So it really depends. But I was working on a particular kind of lipid that had been found in rocks going back several billion years, and it's kind of a bacterial analog to cholesterol, which we all have in our bodies. But bacteria don't make cholesterol, and so there's kind of an enigma or a mystery about why bacteria have this.
How is it called?
Hopanoids.
Okay.
Yeah, and so these hopanoids were originally found in oils and sediments. So basically, any sediment you looked at, you'd find a hopanoid going all the way back to as far back as you can detect lipids and sediments. And no one really knew what they were until the 80s when they were discovered in bacteria.
Okay.
So that was...
And they are of biological origin.
Mm-hmm. Yeah, so you can tell they're of biological origin because they have this ring structure that could only be produced through enzymatic reactions.
Okay.
As opposed to maybe like a linear structure like a fatty acid, which just has carbons, which in principle can be made abiotically. So if you see something like that, then you know that an enzyme must have been involved. And so I was working on trying to understand like what if you find a hopanoid in an ancient sediment, what does that tell you about the environment that was happening back then? And then the thing I got really interested in at the end of my PhD was, can we actually learn something about the complexity of cells that made these hopanoids in ancient times? And so that really required thinking about what they actually do in membranes. And so then I came to Dresden originally to work with Kai Siemens, who is one of the founding directors of the Max Planck here, because he had been working on cholesterol and its role in organizing membranes. And he was one of the founders of this lipid raft area. So I wanted to come and try to use all the tools they had developed working with sterols and membranes and apply them to hopanoids. And I did that, and we found we showed, that in experimental systems, hopanoids actually have some very similar properties as sterols. And so they may have actually, life before cholesterol could have potentially organized its membranes in similar ways using hopanoids.
All right. Do you have any question so far? No? Okay. Then I thought that probably would be a good idea to start with the easy questions.
Yeah.
And by easy, I mean extremely complex. What is life? Since we're going to speak about the origin of life, and I think something that we have to do along the way is to try to define as best as we can what are the properties that a certain organism needs to have in order for us to consider it alive. And where's the line that divides? I know it's a rather fuzzy line. It's very hard to actually trace in a very sharp manner. That divides what's alive from what's not alive. You already mentioned one, at least albeit indirectly, one of these properties that I think an organism must have, which is when you're talking about membranes, basically what you're summoning there is a type of structure that will compartmentalize the organism.
Right.
So this whatever thing that we're going to talk about will need to have a certain demarcation, will have a certain physical limit, certain structure that separates it from the rest of the world. Yeah. So perhaps this dividing, something that divides the organism that sets it in a finite amount of space. It's one of the requirements that you would need to have for something to be alive. But then there's I think it's the discussion continues about what are the other features that organisms must have in order to be alive. And one of the typical, I would say, examples that are continually raised when we're trying to define this kind of thing, what life is for instance, the example of viruses. Viruses are a type of organism, a type of structure that for the most part are rather inactive. They are not doing much. They are basically a strand of DNAs cased in a very fancy box made out of proteins. And if they are not in the right type of environment, they are just basically inert. They're just waiting to find themselves in the right kind of situation, meaning near a cell that they can invade and they can hijack. Yeah. Once they penetrate the cell, this case that surrounds them is very specialized in penetrating a specific type of cell. Yeah. They go directly for the nucleus, and then they also drill through the nucleus, and the case basically opens, and the strand of DNA that they contain inserts itself within the DNA of the nuclear DNA of the cell, right. And then they essentially hijack the machinery of the cell to kind of make copies and copies of themselves until the cell physically cannot withstand the amount of copies of viruses that it contains anymore, and then the whole thing breaks, literally, basically explodes. They go into invade another cell, and then this is there's a very, I would say, sharp contrast between how viruses spend most of their existence, which is just sitting around doing nothing. Yeah. They don't have a metabolism, for instance, or they're not doing much. And then they kind of shift into action once they find themselves in the right circumstance. Yeah. So the question is that a kind of organism something that you would say it's a living organism or not? Or which properties is it lacking, or which properties is it? Yeah.
Well, I mean, it kind of comes to a similar question, which is, recently the Craig Venter Institute produced this minimal cell. Yeah. There was a paper a couple of years ago, and they basically took a really simple bacteria and they knocked out as many genes as they could to get down to kind of a core genome of about 500 genes that they said was, okay, this is a minimal organism that's capable of growing. But then you have to ask yourself, okay, what do we want the minimal organism to do to call it minimal life? So in the case of this organism, it's a bacteria that, for example, uses sugar for its main energy source. and it requires a lot of. It can't make, for example, a lot of amino acids or lipids or things like that.
So it has a metabolism, for instance.
Yeah, so there are elements that there are structural components of the cell that it actually can't make itself that have to be fed to it, because it's derived from basically a bacterial pathogen that lives in mammals that gets all these things from the host, right?
Yeah.
So then you could say, okay, well, that's a minimal cell in the context of an environment where you have all these ready-made parts, right? So then you have to ask, okay, well, what do we mean by minimal life? Do we then want instead something that's a minimal autotroph, something that, like a photosynthetic minimal cell that could survive entirely autonomously?
Yeah.
So, I mean, these are all valid. I think there's no right answer, right? It's just where do you want to put
the draw the line.
Exactly.
So this minimal cell can divide as well?
Yeah. They grow. I mean, they're a little slow. They're not happy. We work with them in the lab. So, but and this also, goes. We can get back to it later. But I think when you get down to how life started and what kind of systems were emerging, there must have been at some point a dependence on the source of building blocks and things like that. So,
yeah, I mean, I think the reason why this question is relevant when you want to investigate the origins of life, it's because you need to at least kind of isolate or construct, let's say, a rough list of the ingredients that you would need to have in place.
Yeah.
That would need to be in the environment where the first signals of life arose.
Yeah.
So they would. I think that's the utility for the most part.
So I think in thinking about what are your thresholds, what do you want to? I mean, that's for sure. The standard things that people think about when you want to define life, for the first thing I would think of would be replication.
Yeah, that's quite common. Yes.
Yeah, you need a way to transmit information from one generation to the next. There has to be some kind of selection, so that requires life and death and cycles. So replication is a really important part.
And replication means almost identical replication, or that's not a criteria?
That's not necessarily. I mean, actually, in a way, you need errors and lack of fidelity.
But percentage-wise, that is a very minor fraction of it.
Yeah, but that's up for debate, I guess. Is how much error can you tolerate?
Yeah, it seems like just a nuance, but it's a very important detail, right? I mean, in order forgetting now about the question, just bypassing the question about the origin of life just for a moment, in order for Darwinian evolution to occur and to be possible, you need a system that is not perfect at replicating, right? Otherwise, evolution wouldn't.
You wouldn't have any selection.
So it's just, you don't have any of that. You don't have evolution at all.
No, because you can think of a lot of chemistry, which we will assume as lifeless, which incorporates capsules, which are like compartments, which can divide into like self-replicates. But most people won't call it. Like soap bubbles also, you can give it certain triggers and it will divide into two things, which will in principle be...
There's also the analogy, I think, I don't remember where I read this one, I think it was probably the selfish gene. The analogy with fire, for instance, right? I mean, you have a fire can replicate itself, you just need to find another place where a gust of wind to blow a little bit of fire somewhere else, and you have kind of essentially a replication of the original fire, but you by no means would call that a living system, right?
No,
so that's again going back to the point I was trying to make before, that if you look at any of the single features in isolation, you don't get a living system. You need to have them. Several of them, a combination, and acting in a very orchestrated.
So that brings up another point. I mean, the problem with fire is it can't move and it can't change its environment in order to make the right environment, the right surroundings, so that it can continue.
Yeah.
Right. And so, I mean, one of the other key aspects that life has is it's responsive. So it can adapt to its environment, and it can also modify its environment.
Yeah, that's also kind of in line with the concept of the extended phenotype, which also comes from Dawkins. He also speculated on that. Anyway, when you mentioned replication, there's a replication as being one of these core aspects of living organisms, and what kind of a role it could have played at the very origin of life. Because there are certain, I don't know if I would call them schools of thought or competing views about how life started, and one of them is the replication first. Replication happened first, which is basically also kind of commonly referred to as the RNA world. That probably the kind of system that we had at the very beginning was something akin to replicator molecules kind of floating around, and RNA is one of these candidates probably for the people that adopt this view, is the most probable candidate of molecule, because it can encode information and it can also act as a catalyst.
That's right.
To build other proteins. As far as I know, some of the drawbacks of that kind of theory is that RNA itself is a very complicated. It's a very complex molecule, right? And then you kind of have like a locking, like a feedback lock problem there, which is so the RNA molecule is complex enough that it needs to be assembled in some way, and at the same time, this RNA with its catalytic properties would need to participate in the assemblage of proteins that would then assemble the RNA. It's kind of, I don't know how you break out of this kind of stuff.
I mean, there are people that have done a lot of interesting work. So there are different stages of that problem. I mean, one is what is the chemistry required to get nucleic acid, so the building blocks for RNA and DNA. And there's, I mean, people like John Sutherland and Jack Szostak have made some really important contributions to understanding what's required there. And there's actually some plausible scenarios for how you could, in an pre-biochemical world, get the building blocks for RNA and then have it polymerize and eventually evolve function. So there are actually experiments and that have been done that isolating these different stages that work. But another problem to bring into this is, okay, so we have replication. Say you have an RNA world or something with these functions like ribozymes that can carry out reactions and also contain information and self-replicate. But then you still need you need energy, and you need a system that can harness that energy in order to maintain itself. So that's kind of a third theme now. I mean, we have replication. What was the first thing?
Compartmentalization.
Responsiveness, compartmentalization, and then you need energy. So these RNA world there's a lot of plausible, pieces of evidence that suggest that an RNA world is a reasonable way to think about things.
Yes.
But then you have to introduce a way that energy can flow through that system.
Yeah, flow is a key part there, I think, for the most part. Channeled flow of energy, because there are ways to provide energy. For instance, the RNA world is also compatible with the primordial kind of scheme, right? Which was started in 1956, the classical experiment by
Uri
Miller, where basically grabbed some flasks, added some organic substances, and they kind of made bolts of lightning pass through these gases, and they happen to synthesize amino acids, which are the building blocks of RNA, proteins, etc. That's well and good, but what you get when you look at actually closer to details of that kind of system is that there's no directional flow of energy there. So what you will have is essentially a soup, of ingredients that are kind of a bunch of organic molecules, but they will not do what is required to construct a living organism. Then there are other bigger problems that have to do with, okay, how certain are we that that's kind of the environment that we have in the very ancient era? Right? And even you can even look at even closer at the details and the problems that exist there. The problem of concentration. Basically, we had an Earth which was almost entirely made of water, with just a little bit pieces of land here and there. So
I would say, I mean, I think that any reasonable person would say that we're never going to know how life actually started unless you invent a time machine.
We could stop the episode here. This is it. Right. Bye,
guys. But I mean, that being said, the idea of thinking about origin of life is an important thought experiment that allows us to explore plausible scenarios for how you could build life from scratch. And that's an important question in the next few decades, the next century maybe, to understand what are the special ingredients that allows life to come out of nothing. How can we engineer it for technology and medicine? And really cracking that code and that problem is fundamental. So we'll never know how life actually started, but understanding how it could start is super important. And I think that's the best way to think about these.
But if we can recreate life synthetically, then we've got enough cues on how life could have started.
That's right. Yeah. I mean, you could probably make some reasonable assumptions based on what Earth history
and progress. I mean, there's some.
So we will get more and more cues on what used to be the case. without the complexity.
Assuming it started
of
on Earth.
Yeah, assuming it's
well, yeah, that's another avenue we could explore.
Yeah.
I'm not a big fan of that one,
Yeah.
but because that just kind of moves. It's moving the goalpost, right?
Yeah.
You move you transfer the problem to
somewhere
the
else.
like to somewhere else. I don't know what
So,
the situation over there.
And there are like people that think about this statistically in terms of how likely is it that a living system would evolve if given enough time? Is it a problem that's so complex that it would almost never happen? Or is it something that just happens no matter what if there's no competing other life
That's
form?
a very interesting aspect because if our knowledge about the origin of life is correct, it started relatively early in the history of Earth. So our planet is around 4.5 billion years old, and turns out that after 500 million years or something like that, the earliest.
So I think already
pieces
in
of
the
evidence.
4 billion year states, you have like certain sort of structures formed in some ancient
That's
rocks.
right. So there are these whorls and things that are found and really,
which
really...
they think that must have happened systematically, which they say.
The point is that geologically speaking, is early. It's in the early Earth when that's.
This is before. But the thing is, when you go back that far, the records are very murky. So you're dealing with very few places on Earth where there are rocks that are that old that are preserved, and so there are
Greenland.
Greenland. There's places in Africa and in North Canada. So people are interpreting these shapes, and it's highly debated. But it's plausible that life could have been around then. Which,
But well, something that we can at least say with a high level of security is that there has been life in this planet for longer than there has not been life in this planet.
That's right,
So people argue that or think that maybe the origin of life, it's a complicated problem. It's probabilistically there's small chances of it happening, but not that small, for instance, compared to the origin of complex life, something like the Eukaryote cells.
Also interesting
We
question.
can also talk about that later. So we need to explore a bit more about these theories, these ideas regarding the very origin of life. So I think the part that is baffling, at least to me as a layman in this, when I try to think about this topic, is that how do you pass from a world of inorganic chemistry to a world of organic chemistry and very highly complex organic chemistry, and to start building up these complex structures that are needed for life. So we already talked a little bit, we brushed over it, but about the scenario or the theory of the primordial soup, and we have highlighted a little bit of what are the problems with that kind of theory. It's a very static world. There's not much happening, and where do you get the energy to derive this chemistry?
Flow
that
of
is
energy.
necessary? And flow is very important. Actually, I'm just going to borrow ideas from different people here. There's he's a philosopher, Daniel Dennett. He recently wrote a book called "From Bacteria to Back and Back," Back and Back. And then he talks a little bit about the origin of life, and he says something about like what you needed to have it's a system that is not a living system, but it's persistent. So you need to have a certain system with a certain level of complexity and energy flow that just persists in this dynamic state for a long enough period of time for something complex to arise. And then if you take that and contrast it with ideas from other people, like there's this Russian-Belgian physicist Ilya Prigogine, who is also has dedicated most part of his career to study complex systems. He has this idea of the kind of system that is a good persister is a dissipative system, which some examples of dissipative systems are, for instance, cyclones, right? Cyclones are highly dynamic and they persist for a long period of time. There's the great spot in Jupiter, which is like a storm that has been raging for hundreds of years, and there's no signals that it's going to stop anytime soon. And you have energy flowing there in a specific manner, in a kind of dynamic equilibrium type of state. And then you have ideas from other people like Bill Martin and Nick Lane, Mike Russell also, who offer the kind of system here on Earth now, and also very likely in the ancient Earth, that could have provided just such that environment. And they talk about hydrothermal vents on the bottom of the sea. Now there are at least 2 different kinds of these vents. One of them is the so-called black smokers, which are, if I'm not mistaken, they are created by the interaction between very hot magma and the cold water of the
So
ocean.
these are the ones that form at the very, so you have these spreading ridges on the bottom of the ocean, and these are at the center, and they're the highest temperature system. So you have water circulating down through magma and, coming out at 400 degrees Celsius.
Yeah.
And you have these incredible gradients from 400 degrees to 4 degrees in a matter of centimeters. But those are considered to be less likely as the hosting environments than there are other environments that are sort of off of the axis of the spreading ridge that are lower temperature.
Yeah.
And they have a lot of interesting chemistry that people think might be a bit more conducive to these sort of carbon metabolism.
Yeah. So as far as I understand, probably the main problem with these black smokers environment is that they're just simply too violent for it to be like hospitable enough, like stable enough for the kind of chemistry that you need for life. There was one German scientist, I don't know his first name, his family name is Westerhäuser, who kind of came up with some fancy chemistry because that was kind of like he favored the black smokers as the environment in which life first arose, and he came up with very ingeniously, I assume, in the lab way to kind of use inorganic catalysts, iron sulfides, I think, like that are expelled in these black smokers, and then also there's like carbon monoxide and hydrogen interacting with these iron sulfides, and then you can obtain some organic compounds. But but as you said,
I had friends in grad school that were working on this stuff. They had these great these golden sacks. They use gold because it's not as reactive.
Golden sacks.
Just like this, like it looks like a little, like kind of a little tube, and then they would pressurize them, up to pressures you would experience at the bottom of the ocean. Heat them up to 400 degrees. They fill them with a bunch of basalt and stuff that they get, and then look to see if they get any organic or sulfur compounds coming out and stuff.
And then you have this other type of hydrothermal vents that you also mentioned. I think they're called alkaline. They're referred to as alkaline hydrothermal vents, which are
much milder.
Yeah, and they're formed through something. Again, like this has a reaction called serpentinization, which is the one that is forming these kind of structures. And the key thing there is that they are not like tiny volcanoes; they are just erupting very violently, those plumes and into environment. But they are kind of like very porous, very cavernous kind of structures. And inside they have like tiny like corridors and chambers, and through which this water flows, highly acidic water can flow through there. And the idea is that these tiny chambers that are part of these structures probably provide like a rudimentary template for ancient cells to form. So that
can I ask? How common are these hydrothermal vents?
Well,
like how prevalent are they? Do we see them everywhere?
Yeah, they're pretty widespread. I mean, because you're looking at.
So is it just any kind of hot water springs? They will be around.
Well, I mean, what you have is like, for example, on the Atlantic Ocean, you have a spreading ridge that runs pretty much in the center from ice. It runs through Iceland and then down south. And then the center is this place where you get these very violent, hot things. And then as you go away from that towards the east or west, you have these wide expanses of places that are some of them are not so well exposed. So there was Lost City was one of the first ones.
In the Atlantic.
Exactly, yeah. And that was one of the ones that they found. I mean, it's difficult to find these things because the ocean is very poorly mapped in the deep ocean.
Yeah, sure.
And the technology to get there is expensive and time consuming. Yeah. But they're in principle large surface area of these sort of environments.
I think one of these that I was reading about is ridiculously high. Like these things are massive structures.
The black smokers are
so these are the central. Exactly. These big sulfur things.
Yeah, you don't get the sense of scale when you look at
the
the
alkaline ones can be quite because the scale of time for which they have existed is like millions of years at least, or even billions of years.
But something that surprised me a lot when I first saw photographs of that, I mean, I saw the structure and I found it there. It was amazing, but I had I couldn't feel a sense of scale of how big these black smokers are until I saw another picture with like a little submarine in between. It's just these things are, like 5-story buildings or something like that. So it's just very impressive structures. Yeah, sure. Don't worry, I take care of this. You took something. I'm sorry. There's something I want to say about that, but no.
But that's the key thing here, right? Like when I talk about the timeline of it, with the black smokers, the timeline is not enough for them to really because they grow and then they within 500,000 years at least at the max, they kind of go down. With the alkaline smoker, the alkaline vents, they can last for millions and millions of years. So potentially, that's also a cue that comes into place.
Yes, so there are several. The reason why they were encouraging in the beginning is because when people discovered them, they found like very rich ecosystems. I mean, actually, Nick Lane in his talks, he compares them. He says that density of life in these ecosystems is as high as in the rainforest. No, in the black smokers.
In the black smokers,
so they are these animals that I've never seen before. Giant tube worms without mouth and without a name.
These things are amazing.
And like eyeless shrimps and God knows which other kind of creatures. So that was very encouraging. People never thought, would find this.
But sometimes thinking about where life started, the environments where life started may not necessarily be the environments where life now exists.
Absolutely.
Because once it has evolved, complex life has evolved, it can sustain itself in a lot of environments which is not even conceivable.
Exactly.
Yeah, that's also now that you mentioned that, there's also I don't remember. I think you used to say Sutherland. What's the name of these?
Sutherland, John Sutherland.
Well, maybe I'm mixing up the names and the experiments, but that he thought that this environment, again going back to the primordial soup, that one of the possible sources of energy could have been UV radiation, and that some of the organic molecules that must have been there were like cyanides and cyanamides and stuff like that. And one of the objections today is that any living organism existing today cannot deal with cyanide, right? And cyanide is toxic for them, and UV radiation is very destructive to life, rather than constructive.
So that's actually another debate as to whether the chemistry there's different starting points. One is, where do you get the building blocks? You have to initially synthesize all the stuff to make a cell or make a system that works, right? And then you have to think about how it could have worked and organized and stuff. And one of the questions is, did these building blocks come out in an environment that would have been conducive to life, or in chemistry that would have been supporting life, or did they form in a completely different hostile chemistry environment and then accumulated and then later would have been around to allow these things to happen? I think they're both open. These are really two camps now. I mean, you have sort of John Sutherland and Shostak and these people, and then you have Nick Lane and Bill Martin. In a way, in terms of their arguments, they're kind of you get the sense from the way they debate it that there's sort of mutually exclusive ideas there. But I think in reality, you really have to think about these are not mutually exclusive ideas. The UV, for example, I mean, this is something that there are actually a lot of research showing that UV is specifically the wavelengths that aren't filtered out through the atmosphere can promote the formation of iron sulfur compounds in shallow water systems.
And iron sulfur compounds are important because they can be the primitive catalysts to make reactive carbon.
Right.
So unless you can make carbon to be reactive, you cannot form these building blocks.
That's a very important point, because life must have started from inorganic chemistry. Inorganic compounds.
So one of the really interesting things about Bill Martin and Nick Lane's story is that they go to these environments in the deep ocean where you have iron minerals and iron sulfur minerals, and they show that, of course, iron catalyzed chemistry is really important in our cells now. A lot of enzymes have an iron component, iron sulfur core, and so their idea was that life maybe started with minerals that were catalyzing these reactions, and then eventually it co-opted these minerals and made it portable, so that cells could go away with it. So that's a really important point. I think that's one of the strongest aspects of their story. But on the other hand, I mean, you have to keep an open mind, and of course, there are studies showing that you can get these kind of chemistries, iron sulfur structures, from for example, UV exposure and things like that. So, it's
also another thing that is very, I would say, compelling from the story is that, again, the environment seems to be kind of ideal because you have this very robust template, which are these pores in the alkaline vents.
Yeah, that's right.
And they are like surrounded by channels where water flows relatively gently, and this is acidic water with lots of hydrogen.
Yeah.
And then you have like this natural proton gradient, which is very important for essentially. There are some exceptions, but it's almost universal to life.
Yeah.
There's the proton motive force, which was the hypothesis formulated by Peter Mitchell, I believe.
Yeah.
That turns out that it's one of the, almost as the genetic code, it's almost universal to life.
That's right.
Life is powered by this proton
There are
motive force.
some
fermentation,
which use fermentation.
Yeah, so those are the exception. But other than that, other organisms for respiration use the proton motive force to produce ATP, to produce their energy.
So the energy of all life is driven by these gradients of hydrogen ions.
Yes.
And the point is there. The point I wanted to make there is that these inorganic cells in these alkaline vents are naturally have that proton gradient.
Yeah.
So there's this water flow outside where concentration of protons is high, and presumably inside the concentration of protons is lower. There's this very thin...
Plus water back in the primitive, in like 4 billion years back would have been more acidic.
So that's the thing. And oxygen was not plentiful, but carbon monoxide was more plentiful in the ancient ocean. And these inorganic cells have very thin layers of iron sulfide compounds, which are the catalysts that could promote the formation of the first organic molecules. So you have it's obviously, as you said before, I mean, to have hopes that we are ever going to get the exact right answer and the exact right kind of we'll know the full environment and how it'll happen step by step. It's probably asking for much, but these elements being there make the argument compelling.
And I mean, one of the things that comes out of so you're talking now about in these vent systems, right, that you have some kind of spatial gradients. So you have gradients of hydrogen ions, and this is what now most life uses to generate energy. This brings up another really important point about what's required for life and what is life. One of the key things is organization. So organizing things in space and in time. And I mean, we can sort of make a rhetorical scenario here. You know, if you look, there's the whole field of synthetic biology, especially this particular area of synthetic biology that's trying to build things from the bottom up. Is focused on trying to make cells. Essentially, there are people like Kate Adamala in Minnesota who's doing. She actually gives some really nice TED talks, and she's doing some fantastic work trying to build up proto cells and stuff from parts, right? And but up to now, no one has managed to really build a robust self-replicating system from basic components. And despite the fact that we have, we know the genetic basis for the code for life. We know what that code makes and what the parts are that are made by that code. We know how many of those parts interact to form metabolism and networks and stuff. But still no one's managed to take all these components, put them in a tube and have a cell come out. So one of the really important missing pieces now to understanding that problem is how does life organize itself? What are the fundamental organizational principles? And of course, in this vent system, part of the organization that alluded to is the spatial organization of having a gradient that's templated by the mineral structure. But really, if we would just back off generally, then I mean, in any environment you think of, ultimately at some point you can produce biomolecules. You can have energy or a source of energy around, but at some point you have to explain how all those things were put in the right place at the right time.
The self-organization aspect of it. Again, basically you're highlighting that it's not just about having all the ingredients. It's the recipe needs a certain type of like temporal evolution happening in a very orchestrated manner in order to get the end product, which is a living organism. It's not just about mixing everything up and then just... although...
And does the other camp of people have ideas on that basis?
Well, I think at this point that's still a really unexplored area because a lot of the attention has been on trying to understand the chemistry. How do you even get the building blocks for life in the first place, or the plausible scenarios, and then energy, and also thinking about what are the plausible environments where all this could have emerged. But really, I think now there is a lot of attention now being focused on organization. I mean, if you look at, of course, I mean, when I came to Kai's lab, he was working on lipid rafts, and the concept was that membranes and modern cells, of course, very complex systems, but that they can organize themselves using sort of the interactions of the components of the membrane, lipids and proteins. And that was really transformative. And then now there's a lot of work also from Dresden, Tony Hyman, working on understanding how phase separation and the cytoplasm of the cell can act to organize biological
So even without
systems.
membranes, you can compartmentalize things into a high concentration.
Without
phase.
membranes?
Yeah, sure.
Yeah.
Oh,
that's
wow.
right.
Yeah. So it's basically having having a valley between two valleys between an energetic barrier.
Wait, I'm not picturing this. So you have like a solution.
Yes. So if you think of oil and water and you mix them,
Alright.
and the oil kind of phase separates from water.
Alright,
So it's something like that, because oil molecules like oil molecules and water molecules like water molecules. So now if you have proteins which is in a very low density concentration,
They form
you can form these kind of
networks and kind of thing. I don't know.
not networks, it's just weak interactions between them where they favor each other in comparison. And this is what in Dresden right now a lot of people are getting into.
Oh.
And then when you concentrate things, then you can have reactive chemistry in these concentrated.
Right. So it comes down to basically creating different compartments and being able to control when certain things go to one compartment or the other. And this gives you the possibility to have cycles into
But that
So this is membraneless compartments, which would so if you think of bacteria, they don't really have membranous compartments within the bacteria, right?
there's no organelles like the mitochondria. And
But the compartmentalization and the organization is happening in terms of energetics and interaction between different types of components in the cell.
So I think that's a really interesting area that I know nothing about, but but studying sort of the fundamental physical principles involved in how you can get organization. And I mean, we're working on some aspects of that with membranes, of course. Membranes are an important element of organizing things. People think of them as compartmentalizing, but they're not only just encapsulating things, but they're also organizing things within the plane. And then, of course, you have these systems in just three-dimensional fluids, like the cytoplasm, that can actually phase separate, like oil and water.
And another aspect which I find fascinating about membranes is that the possibility of having, or the transition from inorganic membranes, supposing, again, that we have this scenario in which we have these hydrothermal events, the transition from inorganic template membranes to biological organic membranes, it's something that allowed these ancient life forms to spread and to move away from the template in which they were created. So then, essentially, they can now explore the world and literally conquer the world. They can just go around, move.
Yeah, exactly. And that would have been a crucial step in the evolution of life, would be to imagine you have some environment where whether it's a puddle or a hydrothermal vent. I'm unbiased in that. But at some point, then the system has to be able to move away from that. Otherwise, that system will go away and it'll disappear forever. So clearly, having a vessel that you can go away and
No, but of course you buy that. But one thing that we constantly miscalculate is the amount of time that existed, right?
Yeah.
So there must have been millions of tries before something succeeded.
Look, if you say the Earth is 4.56 billion years old, and if the first evidence of life you believe in is 3 billion years, let's be conservative or whatever, or let's say or 3.5, 500 million years or 1,000 million years is a shitload of time.
Exactly.
I mean, imagine the scale of a bacteria divides in the E. coli that takes 30 minutes to divide. A really crappy bacteria maybe would take I used to work with these marine cyanobacteria that took a month to grow up to dense culture.
Oh boy.
So you have like maybe a day or two doubling time.
Those are crappy bacteria.
They're really crappy, but they're slow. But even on the scale of 1,000,000 years or 1,000 years even, That's nothing. So you can do, I mean, you can think about evolution as simply a computational problem. Every time you have a division and selection, that's one experiment, that's one piece of processing that happens.
Exploring a pathway, this possible pathway, see how far it takes me
exactly.
to work, okay.
And if you think about, I mean, the amount of computational power associated with 500 million years of repeating that experiment is huge. I mean, it's we have nothing. I don't think we have anything comparable today. Now, I'm not an expert in that area, so maybe you should check that. But I would imagine there's a really staggering amount of power in having that length of time to keep trying things. So even that short period of Earth history, is an amazingly long period of time to have something pop out.
Yeah, which is it's not the short. I mean, I'm not going to be as conservative as you are. I'm going to say probably life originated after 500 million years or so. Probably those are the beginnings of that.
Which
I know that's debatable. But sorry, I'm doing this on purpose because I want to point out that for at least the first 2 billion years of the living organisms being around, you have only bacteria and archaea around.
Or something. Yeah.
So, but for the most part, you have what we now refer to as not complex life compared to eukaryotic cells.
Yeah.
And that's all you get.
Yeah.
And
for 2 billion years.
Yeah.
Yes.
But actually, if you think, so if we look at, I mean, the thing I was studying, these hopanoids, those are found pretty ancient sediments. And there's some debate over whether we can believe all of the cases where they find them. But let's say, 2.7 billion years, 2.5. 1.7 is the best accepted. Yeah. But if you want to get a little bit shady, you can go farther back. But you have something that makes these kind of lipids, suggesting that those organisms were more or less along the same level of complexity as modern bacteria. I mean, they had the pathways to make a fairly complex lipid, right? I mean,
except that it didn't use oxygen, but something else.
Right, right.
So it tells you something about the environment back then.
That's true. That's true. But the one of the interesting things there is also that imagine, already very early on you have cells that are almost as complex as modern life. Right. I mean, and if you look at bacteria, they're not that simple.
Sure. When I use the word complex here, I'm using it in a very relative way.
Yeah.
I'm just trying to emphasize kind of the leap in complexity that you find between bacteria and archaea and eukaryotic cells, which are the cells that form complex and multicellular animals. What I understand that the bacteria are just by no means simple.
Yeah, I mean, this is all bullshit, right? But I mean, my imagination is that the step from no life to a protocell was probably a much more complicated path than the step from a single cell organism to a multicellular organism.
From
single cell to multicellular,
or
even complicated eukaryotes.
Yeah, or from prokaryote to eukaryote.
Yeah, because at this point you have all the building blocks. You have a lot of genes. You have a lot of pathways, and at this point there's a lot of reworking, and there's building up on things, right? But getting the first, look at E. coli. I mean, this is something that's fantastically complicated. Yeah. I mean, no one has even managed to model computationally how something like that would work.
But that's a big
So the argument with why complex life might also be a massive jump is because it has only happened once. Like you don't have many lines of something similar to eukaryotes happening over
That's
time.
Also true.
So there's a monophile
from
I think what's crucial there is
over a period of 4.5 billion years.
Yeah.
So that's the only thing
I think what's crucial there for you to decide this matter, to try to decide what is more difficult leap to accomplish than what is if you have the conservative approach to when life originated, then probably there's a 500 million years of only bacteria being around.
Yeah.
But if you are more towards the other end, where you think that life probably originated after 500 million years of the Earth being there, then you have nearly 2 billion years of only bacteria. So you have 2 billion years of bacteria, and then only then the jump to eukaryotic complex life happened. Then you would probably have to say, I think it's more complex. It's less probable the formation of eukaryotic cells than the origin of life itself. And if you are on the other extreme, well, then it seems the other way around.
Yeah. But Yeah, I mean, but on the other hand, also the last common ancestor we only have one example of life emerging, right? So I mean, that's one of the. I mean, there was the arsenic life paper back in 2010. You know, I mean, there are people looking for other forms of life that could have emerged in parallel. And no one has yet found any good evidence for something like that on Earth.
Can you elaborate a bit more about this paper? You were telling us off camera a little bit of the story, and I found it
it was
a very warning story.
Classic disasters in science. It was a paper that came out, and it was a young scientist who was thinking about trying to find alternate forms of life, and her idea was.
And by alternate form, you mean like other nucleotide.
Exactly.
Possibilities.
Something that would have
DNA or RNA.
Genetic code in this case.
Yeah, and so she was looking for things that had a different genetic code or a different basis for information. And one of the ideas was to look for something that replaced. It used a different chemical composition for the DNA. So phosphorus is one of the main components of DNA, and arsenic is an element that has a very similar chemical property. In terms of the two elements, phosphorus and arsenic have very similar sizes and properties. So she was looking for things that maybe had arsenic instead of phosphate. And they went to her and this team she was working with went to this lake that was basically this toxic lake full with arsenic. And they managed to isolate a bacterial organism that can grow in this environment. And they did some experiments, and what they were trying to do was determine if the DNA had incorporated arsenic, and if it. So they published this paper in Science, and the conclusion was that they had found an organism that replaced phosphorus with arsenic in its DNA backbone. And immediately, as soon as it was published, people were very skeptical because it's a big claim. And then
turns out they should have been more skeptical.
They should have been more skeptical because people started looking at the paper, and it turns out they just didn't do the proper controls, and they didn't actually make the measurements that were necessary to really show that. And so it turned what they really measured was kind of noise. I mean, it wasn't. I won't get into the details, but there were a lot of. So then, instead of it was interesting, and Science didn't retract the paper, but they published in parallel a bunch of other studies that. The scientific community is really amazing sometimes. People got together and they said, "This is a really controversial topic," and some people dedicated a year of their lab time, to actually try to reproduce some of the results and explore why it might or might not have been right or wrong.
No, because if it's true, it is amazing, right?
Because
exactly. You have to really understand the structure. There's so many questions that pop up. As soon as if it's something true, right?
Right. What would be the selection for that?
And this has huge implications, even if you want to think about life at a universal scale. For instance, does life has to be anywhere else? The same, exactly. Does it have to evolve and happen in the same way in which it happened here? Or this is just really a thing of chance and our specific circumstances?
So unfortunately, in this case, it turned out that they published all these things, and it turned out it was just a very arsenic tolerant organism, and if there is any replacement, it's purely accidental. Because there is some non-specificity in the how they build the DNA, but also then you have to ask, okay, is this really a different form of life if it still uses the same genetic code?
No. I talking about complexity again. While I said that eukaryotic cells are commonly regarded as way more complex than bacteria, because the appearance of the eukaryotic cells allow the forms of life to grow much larger and eventually to form multicellular organisms and explore design space in a very kind of widespread manner. On the other hand, bacteria became like the masters of biochemistry complexity, biochemical complexity. So you can find bacteria living out of essentially anything. They have mastered the ability to subtract energy from almost any kind of chemical compound you put in front of them.
So both of you are talking about the same. So there is a jump to life, but there's also a jump to eukaryotic life. That's the argument. It's not like the eukaryotic jump is more unlikely than the.
There is some idea that one of the limiting factors could have been oxygen. So I mean, when you have a tissue or a multicellular thing, you have to think about some of the essential element, like nutrients, getting to the cells that are at the core of this structure. So you have a thing, and so for multicellular life that relies on aerobic metabolism that requires oxygen.
Aerobic means with oxygen.
Exactly, with oxygen.
Respiration.
then you have to think about what would be the minimal oxygen pressure in the atmosphere, the amount of oxygen in the atmosphere required for a multicellular organism to actually be able to support all the cells.
Yes.
And so there is some debate, or there's been discussion about whether this point when oxygen began to rise because some cyanobacteria evolved, and those plant basically photosynthetic organisms that produce oxygen oxygenated the surface of the earth, and when that happened, it suddenly became possible to support a multicellular system that uses respiration.
So there again, we're talking about several leaps in terms of the history of life, right? So the first one is obviously the very origin, and then the second one that looms very prominently is the shift from prokaryotic to eukaryotic life, and then from there from unicellular organisms to multicellular organisms, and going back to focusing for a moment on the jump from prokaryotic to eukaryotic. We know quite a bit about how that might have happened, and the hypothesis, the dominant, as far as I know, actually, well, I don't quite dare to say that it has been proven, but I think it's very, very well established that this must have been a symbiotic event between two different types of cells, an archaea host and a bacteria. How do you call it? The symbiote. Joined the
the symbiosis theory is definitely supported. I mean, the mitochondria clearly derived from a bacteria.
And also the chromat. What's the other thing which for photosynthesis?
The thylakoid.
Anyway,
the plastids.
But this is also one
chromoplast.
This is also the chloroplast.
Chloroplast.
So the the chromoplast. No, let me open another one for you. I feel like you need a little bit more. I want to say something. I want to say something, and I forgot what I wanted to say. This chromoplast totally occupied my mind.
Well, so the symbiosis theory is well supported. I mean, Lynn Margulis was.
Yes, that's what I wanted to do. Because it's a curious story, right? I mean, there were ideas already kind of floating around before, but she was the one that kind of made the whole hypothesis way more robust.
But she argued that pretty much every globular thing that you see inside the cell is through symbiotic.
She had the hypothesis of like a series, like essentially,
So the idea basically is that a bacteria somehow at some point got into an archaea and formed the mitochondria, so mitochondria is what generates energy in the cell.
Where respiration happens.
Exactly. And similarly, plants at some point later on in the stages of evolution got another set of bacteria in there, which allowed them to do photosynthesis. And her argument was all this complexity that you see in regular cells now is not through incremental evolution, but as a process of at different time points, some sort of a symbiosis event.
But you know that she had like an ideological drive towards that theory. So she mixed the thing that is very curious there is that she had plainly a feminist agenda in her scientific thinking. She thought that this was a more feminine way to evolution, and it was in opposition with the masculine way in which natural selection operates. And she said that openly. This is not me making up.
So it's not survival of the fittest, but
it's cooperation. It's emphasizing the cooperative aspect of life at the micro scale. So you have this series of symbiosis events when different bacteria come and join each other, and they help each other, and they enter this like interlocking relationship of mutual benefit and so on. And she openly said that she liked this also because it was a more feminine approach to biology in contrast to the.
But I think that's also an important thing when you think about how scientists work. We're humans, and we have ideas and biases and passions about the ways we think about things. And I think it's one of the that some of the best scientists, the people that have been the most influential, have had strong passions or feelings about how things are, and they push those ideas.
Stubborn.
You have to be stubborn, exactly. And someone like Lynn Margulis was notorious for being a very unique individual. She I mean, people that knew her knew that she was a very eccentric strong-minded thing, and that's one of the personality types that gets ideas like that across. Is you have
to be persistent.
Exactly.
Yeah.
So and sometimes when you're right, then it pays off.
Yeah, I mean, she was definitely right in kind of pointing out the origin of mitochondria.
Yeah.
That mitochondria come basically from alpha proteobacteria.
Yeah, it's interesting. I mean, like now, if you think about there's a lot of interest in the microbiome now. And how bacteria and humans interact. And that's like a big research tool. Everyone puts that in their grant because it gets the money.
No, but it's really really crazy. There's this book. I don't know if you've read it, by Ed Young, called "I Contain Multitudes."
Not yet.
No, it's amazing. I mean, it kind of shakes. It really shakes your worldview. In the way you see multicellular organisms.
No, but
changes radically.
I think he gave a TED talk also.
Well, I don't know about that. Probably it's there, but if it's there, I'm sure it's something it's worthy to check it out.
It's easier to get people to read, to listen than read these days.
Yeah, exactly.
It gives you another very unique sense of scale of the kind of organisms that we are, and how many biodiversity is contained within us. If you grab a sample of the bacteria that live in your gut and contrast that with the bacteria that live in the palm of your hand, it's like a completely different microbiome. Yeah. And then you compare the right hand to the left hand, and maybe there's also going to be significant differences between this. It's really crazy.
Yeah. And then they did these experiments. I think there was one episode in Radiolab where they made two people shake hands. I think one of them was Neil deGrasse Tyson or someone.
No.
And then they see that how much your bacteria is coming into my hand and how much the other way around.
Yeah.
And they found that some are more exploratory than others and stuff like that.
I saw. I mean, so one of the things is you wonder whether this sort of symbiosis idea can constantly be happening. Will there be more cases of new symbiosis with other eukaryotic organisms? I mean, I saw a talk recently at a conference where they were talking about there are pathogens that infect us, and most pathogens don't go into our cells; they stay on the outside. Yeah. But there's some of these bacteria that actually penetrate our cells. They go inside. They live inside of our cells. They like viruses. They replicate and they come out. And some of these have actually acquired genes from us. So they've taken our genes and they use them to hijack the cell machinery. Yeah. And one of the bacteria one person was talking about, they had taken some of the machinery required to hijack the vesicular transport pathway, so they could actually, tag a ride on the these transport mechanisms, so that they could move around the inside of the cell. So I mean, things like that are probably at the starting point.
You get this kind of fractal view of life somehow. Like even single eukaryotic cells are microcosms for smaller bacteria to live. Yeah, exactly. Giardia is one of these little organisms.
No, but the question now is, so this transition from the symbiosis to being completely incorporated within the cell, so that you lose most of your regular activity, and let the bigger cell do it, and you just perform a certain role.
Right.
Like that's a huge jump, and that in principle, like if you look at life, has only happened twice.
Yeah.
And that created a lot. There's this other book also by Nick Lane. I think I forget the title. It's like Sex, Power, and Suicide.
Oh yeah. I think that was his first.
The mitochondria and the meaning of life.
Right.
And all the kind of repercussions that this initial symbiosis had, the origin of mitochondria, in all sorts of aspects of life, it's quite impressive to think about. Like for instance, as you mentioned, so the bacteria that joined the archaean host had a bacterial-sized genome, right? Both had similar-sized genomes. But if you look at today, and you compare the size of the genome of a mitochondria with the size of the nuclear genome, they are largely, hugely different in sizes. The nuclear genome is much, much larger in comparison to the mitochondria. So that is telling you that a huge part of the mitochondrial genome, or the original bacterial genome, had to migrate towards the nucleus. Yeah. And then there has to be like a issue of compatibility between the nucleus. So you had like this compatibility within a single eukaryotic cell, which has to be tuned very, very precisely, because as you mentioned also before,
Any
respiration.
step, it can just
But it's very crucial. It's respiration happens on the membrane of the mitochondria, and for respiration to take place, you need to assemble these very complex respiration units.
Yeah.
But turns out that these complex units are formed by proteins, some of which are encoded by the genes in the mitochondria, but some others are encoded by genes in the nucleus, and they need to fit perfectly, right? If they don't fit perfectly, if there's any mismatch, then the cell is basically not viable. Respiration cannot happen. Yeah. It's the level of precision is just mind blowing, right? In this respiration.
Yeah, but the precision is over time.
Yeah, sure.
So it must have been a much more chaotic event to start off with.
Yeah, but it tells you about the tinkering power of evolution. How precise it is over long periods of time.
So let's just put some context into why we are even arguing about this, right?
Why are we arguing about
this? Good point. Why are we arguing about
this? The whole point is so what Nacho is talking about is how complicated it must have been for to have some sort of a regulated energy source within your cell who is always powering the cell around, which led to this other level of complexity. So the question is, if for instance now we look at stars all around the universe, right, and then you find these minor cues, okay, there's hydrogen there, there's carbon there, there's oxygen there, and now to what accuracy or what confidence we can say that okay, there might be at least something similar to prokaryotic life there, and so if the jump from prokaryotic to eukaryotic and complex life is inevitable, then we can expect that there's a lot of different complex life around us. Yeah. But if that jump is very bleak, then we know that even if we are seeing, there might be a lot of life around in the universe. Yeah. But it is a very, very high likelihood that is just primitive life.
Well, one of the things we're looking at is that once you have something, for example, once you evolve life's pretty competitive. It competes for resources and for an environment and stuff. Yeah. So any other thing that's not life yet that's trying to become life, right? I mean, I'm giving intent, but obviously
it's fine.
But once you have a thing that's taking up the environment, then it's less likely that something else is going to come out, right? So again, anytime there's a niche and you fill that niche, then you have a competitor there, and the chance that something is less prepared is going to come in and displace it is very low. So then you have no life. So
the fact that you have only one line of life, it might be it's just it's like there's only one Homo species, which is Homo sapiens, right? Because we have kind of outcompeted all the other.
Exactly. You look at the laboratory. If we're trying to do an experiment where we make have some bottom up system that we were trying to get a replicase to work or whatever, we constantly have to worry about bacteria contaminating our sample. Yeah. Right, because they're really good at eating all the stuff that we're building, making these experiments from. Yeah. So that's exactly the problem that the things that haven't become life yet are facing. Is that
so life is all about scavenging.
Yeah, when competing for these niches,
It's a brutal picture of nature ready tooth and claw. It's kind of you have to compete. If you have got to compete for limited resources, then there's just no.
So when there's no life, there's no competition. So all kind of exploratory pathways are given some leeway.
Exactly.
But once there's one thing that is out there,
it sets the rules basically.
What's your position? Do you think life is relatively common in the universe?
That's my feeling. I think that's probably a question that's better left to physicists and mathematicians that can assess the but it just seems...
but it's people like you who would say how likely it is. No, not people like you, but Sutherland.
I think the physicists are very cavalier.
They come up with very empirical starting points.
That's true. That's
to start off with.
I think biologists probably have. If I were to ask somebody, that would be a biologist, I guess.
Yeah. Well, clearly it's possible because we're here. And so that means that it's above some threshold where it has to happen at some rate, right? So, and then, the argument always is you look at the universe and, that's one of the areas now that astrobiologists or astrophysicists are working on is identifying planets like Earth that fall in the same kind of category as Earth. and they use all these spectroscopic methods and high-powered telescopes and stuff. And that's one of the questions: is how frequent do planets like this exist? I guess it's a bit of a narrow set of questions about how, because we're saying, okay, how frequent could life like ours have emerged? But then there's a whole other range of crazy, like it gets into the maybe more pseudoscience sometimes. But what if you want to redefine like more broad definition of what life is? Maybe there are other ways you could think about what life is and look out into the universe with your telescope and say, okay, what should we be looking for besides carbon-based?
So this is another debate, right? I mean, people like Nick Lane, for instance, think that if we're going to find life anywhere else, it's not going to be radically different than the life we have here in terms of like it's going to be organic, it's going to be carbon-based, and there's other people that think maybe you can make life out of like silicon-based life, which I find very unlikely. But it's been an argument that has been raised also before. In terms of like, for instance, you can even look at finer details. Is the genetic code universal? Or something like a genetic code universal? Do you need that kind of like template to kind of encrypt information?
These are fascinating questions. I mean, I guess the limiting factor is thinking about what would you even go look for, right? I mean, right now you have people with telescopes and they look at signatures of light and stuff like that. But
if for instance the way life arose and evolved on Earth is representative, then you could, for instance, look at a planet that has an oxygen-rich environment. Because large part, the reason why we have an oxygen-rich environment is the action of life on Earth.
Taking advantage of light energy.
But if oxygen existed in the beginning part of life, it would have just burnt everything. Like you need a certain amount of complexity after which oxygen has to arrive.
But what I'm saying is, if the pathway that life followed here is representative, then maybe an encouraging sign would be to see a planet, analyze the spectrum, and see it's a it's an oxygen-rich environment over there.
So I think the only thing we can say is like give a number for or a range for likelihood. On how likely it is. At the moment, at least.
It seems like it's pretty likely. I mean, we're here, and I mean,
it's a very like it's not anthropocentric, but it's an earthocentric worldview to think that this is the only time it happens.
It's organocentric.
Organocentric.
It just seems impossible to think that this is the only time it's happened. You look, I mean, the more we learn about the universe, I mean, in the last few decades, it's become clear that there's a shitload of stuff out there.
I'm on your side.
I totally believe that life exists. Around the globe. Around the universe. Beyond. But I'm not so certain about complex life.
Complex meaning?
Complex means...
Eukaryote.
Okay, let's say conscious life.
Boy.
I went one more
time. The ladder of complexity just keeps on growing.
Well, I mean, all of these things, at least as far as we understand, right, from Darwinian, the natural selection, is that there has to be some selective advantage, right, for something to emerge. So multicellularity, there was some advantage. Consciousness, there was some advantage. It gave us the ability to make tools and to outcompete predators and things like that, right? So, I mean, you just have to think, where would there be an advantage? And as soon as... I don't know. I mean, consciousness, like, once you have life, then it's going to keep trying to, outcompete each other.
It might be... So okay, let's talk about multicellularity. Because I think if multicellularity exists, then maybe it's a common thing that just pops in.
I think one related way to think about this is thinking about this in terms of convergent evolution. So just to cite an example, the vision, an organ that is able to detect light here on Earth, how many times has it evolved independently? Many times. I think it's over 16 different times.
A ridiculous number of different types of eye evolution from cells that can detect light.
Because there's a certain feature of the environment that can be taken advantage of at a certain niche. And if life has this competitive spirit to it, which it definitely has if it follows Darwinian evolution, then very likely you will get these different pathways converging towards exploiting this resource that is available. You have wings on, you have the way bats evolve wings is different, but from the way that birds evolve wings, different from the way insects evolve wings. etc. So, and this can be taken to the extreme. You know, there's this Simon Conway Morris, who believes that actually this is a very powerful force in the evolution of life, and that, for instance, humans are inevitable. He took it that far. He says that if you look at life anywhere else, you will not find, exactly like
humanoid,
my twin and your twin and everybody's twin,
right.
but something that looks very anthropomorphic.
Yeah.
And the curious thing about this is that he is the kind of the central character of another book. He's a real person, but he was kind of his contributions of science were written about by Stephen Jay Gould in his book Wonderful Life, and he takes the opposite case. He focuses on the Cambrian explosion period, and says that if you kind of go back and run back the tape and let things unfold,
would they happen the same way, or how would
he goes?
There's so many stochastic, probabilistic elements that it would have gone in a different direction.
He calls in the notion of contingence, basically luck, and argues that very likely life could have been, radically different in terms of like the anatomical and the kind of organs that would have evolved in complex and multicellular organisms would have been
stochasticity. But on the other hand, again, this is a problem that physicists are better equipped to deal with. But this the butterfly effect, or whatever you want to call it, online. I mean, the thing is, when you're dealing with so many repetitions, with millions and billions of years, it seems very unlikely that luck has much to do with anything. You're fully exploring the space you can explore in that.
Yeah,
but now you're arguing in the other direction, right? Like you were talking about how once you have something defined, it scavenges out everything else.
Sure. Yeah. Okay. But
in the initial stages, there's every possible exploratory pathway is available.
Exactly, and at some point,
because you have enough time to do it.
Yeah, and I think the point is that you will continue to reach different levels of complexity.
Yeah,
it's just a matter of how long you have to do it. But I mean, of course, once you get to something, then you fill that niche, and it becomes harder to displace the
so it's about capturing a niche. If the niche is not captured, then there's ample opportunities for everyone.
Yeah, there's no competition. You just keep trying and trying and trying. Things keep happening until something falls into that.
It's a computational problem that is run in billions and trillions of different computers.
Yeah, exactly. So, and that's why I think
the problem is going to get solved.
The problem, exactly. If you have a billion years, it will get solved. It's some you will explore enough space, in terms of chemical complexity or organismal complexity,
design space, structural space, all
exactly.
I think it's still about finding out the probabilities of different likelihoods of different, to find out the fundamental blocks. So one block is to just switch on, lights on, life exists. So that must be a fundamental block. So if that is crossed, then you go ahead. But there might be 4 or 5 different blocks.
This is where I think computational biology maybe has an advantage, because in the lab you're limited by how long an experiment takes. If you imagine an experiment takes a day or a week, it took life a million years to evolve something. It's hopeless. You're not gonna. But if you can find, if you can do these experiments, at least in theoretical scenarios, much faster using computation, maybe you can begin to get a little bit farther with seeing how much space you have to explore to get a multicellular organism.
Because there's a lot of space, there's a lot of time. But if there's a ridiculous probability, even that is a likely event,
right?
Because of that,
sheer numbers make small probabilities happen.
Likely,
everything happens. All right, so we already crossed the 1 hour mark for the quarter of an hour. So I think it's around time to start wrapping this up. Any final thought, final words now this time that you want to throw, or something?
Yeah, well, I mean, so okay, we can kind of summarize a little bit. So in terms of origin of life, my feeling is we're never going to know how it started, but we can come up with plausible scenarios, and I think that's one of the biggest values for people that are working on origin of life. One of the greatest things they can contribute is to really begin to hack away at what are the fundamental principles that makes life tick. And this is something that maybe at face value people think of as being like a really esoteric, almost artistic endeavor. But I think that in the long run, the insights that we gain from these kind of approaches, that all these origin of life, I'm not an origin of life expert, but these people like John Sutherland and Nick Lane, and the consequences of their discoveries may have really profound impacts on technology and medicine and human society. And I think we have to, remember that even these esoteric questions have a really important role in the fate of humanity.
But that's not what is driving you, right?
No.
At the end of the day, it's the question.
And that's exactly what science is you're curious and you ask things.
It's the byproduct that is people are using.
Yeah.
But that's not the question that drives you.
No, yeah. But when you look at it selectively, we wouldn't be here if there wasn't a selective advantage for society to have scientists that are working on these problems. Because clearly
about determinism.
Right.
Yeah.
People working on these problems have, the discovery of the structure of DNA. No one would have predicted that people, at the time when Watson and Crick and Rosalind Franklin and all these people were working on it, it was a purely academic question. What's the structural basis for heredity?
Although Watson did say about the helical structure of the DNA double strand that it's so pretty that it had to be true.
But true. All this was before Schrödinger wrote What is Life, or after?
No, this is after Schrödinger.
Yeah. So he kind of the physicist came and influenced.
No. He kind of said Schrödinger with that book set a certain kind of research type of agenda towards
but
way of thinking about life.
No one would have predicted the impact that would have now on molecular medicine.
No.
The genomic revolution and stuff. Yeah. So I think it's important to keep in mind. So these questions about the vital question, the origin of life, what is life,
very, very special.
Exactly.
I don't know. My feeling as an obviously self-declared outsider and just an avid reader of popular science is that we're gonna crack it.
I would bet we have a good chance in the next few decades of someone figuring out how to throw stuff in a tube and make something come out that's self-replicating. But on the flip side, I bet it'll take us significantly longer to understand why that is.
Possible. Fair enough.
So now, so just a cool question to that. If we can do that, will that be DNA and RNA? Because of course we'll give the right nucleotides. So we'll have to give those components in.
Most likely, because that's what people are working with.
Yeah. So we won't come up with a completely authentic.
And you see what happens. You see what happens when you start working with arsenic. Your career can just go in a big downfall.
Exactly.
Let's not do that again. Okay.
Yeah.
So I think it's time to say goodbye.
Cool. Thanks a lot. It was fun.
Yeah, it was definitely fun. Very interesting. I was waiting for this actually for quite a while.
Cool.
All right.
Yeah.
Bye.
Cheers.
Ciao.
If you like what we do, please help us continue doing it simply by subscribing to our Podbean or YouTube channel, following us on Twitter, liking us on Facebook, visiting us on our website tillthebottom.com, or simply spreading the word. Find the links in the description below. The music you hear in the background is a track called Face to Face, performed by Daphne. Thank you for listening, and until next time, cheers.